Hi everyone. I'm just going to try to give you an overview of the things we talked about in lab on Friday. So two of the topics that we discussed when we were doing the Punnett squares is codominance and incomplete dominance. So I'm going to do a little on both of those, but they are both addressed in my text lecture on my website. And there are videos and animations there also. It is imperative that you go there if you want to know the material for the test. It is also in your textbook, so you could use that also. I don't go above and beyond the textbook, and also the lab book, because you have the information from lab the other day. So there's multiple sources of information on this, but this is on the test. So first, looking at blood types. There are 4. Those are the phenotypes, is the blood type. A, AB, B, and O. What is different here is that A and B are both dominant. Thus, when they're together, they're both expressed and become type AB blood. O is recessive, so you always use little i's to indicate the O blood type. When it comes to A or B blood, there are 2 possible genotypes. They could be homozygous dominant, as shown in this picture at the top left, or heterozygous for type A blood. Same thing with B. It can be homozygous dominant or heterozygous. This picture is sort of just another picture of the last slide, but there are multiple alleles involved with ABO blood types. And it's very important to understand what's the genotype, what's the phenotype, and I also have put up in the course some sample problems that you can do and ask me if you did them right. So this graph, it shows everything that you need to know. I do not go into RH, so do not worry about that. All you need to understand is this chart. Red blood cells all have markers on their outside surface. Those are called antigens because they are capable of causing an immune system response. If there's antibodies that match up to the antigen, you'll cause the red blood cells to clump. Antibodies are proteins that your body makes to protect you. So if you have type A blood, the antibodies that you have are against B blood. And if you look at this picture, it's called anti-B, and the ends, or the catching ends of that star type of picture, they're Y's. That Y is what attaches to the antigen, and it's like a lock and key. The antigen has to match up to the antibody for them to connect. Thus, type A blood can take type A blood because the anti-B antibody will not attack type A. It's only going to go after type B. The bottom picture is showing you the antigens, which I already mentioned. They're on the outside surface of the red blood cell. They're also called glycolipids, as you'll see in another slide. It's a protein and a fat and a sugar that make up the antigen. That's all. You don't need to worry about what makes it up, just knowing that it's in your blood and protecting you and part of your immune system response. Type B blood has B markers on it and antibodies against type A blood. And if you look at the catching arms, you will see that the catching arms on the anti-A match up to the A blood. Thus, they would attach and clump if the B type person got type A blood. AB blood has both antigens and no antibodies. This is the universal recipient because there are no antibodies to attack any of the blood that's given to it. O blood has really no antigens on its surface. It's showing you those little markers. There are markers on every blood cell, but those are not antigens in this case. So the O is bare, but it has both antibodies. So O can only receive O, but it is the universal donor because the antibodies won't attack it. So O can go to A because the anti-B antibody is not going to attack it. It can be given to B because the anti-A antibody won't attack it. Group AB doesn't have antibodies. Thus, O is the universal donor, AB is the universal recipient. This is just repeating what I had said on the previous slide. Glycolipid is antigen. Same thing. When you say glycolipid, you're just saying what it's made of. But it is a protein that is projected on the outside of the red blood cell. All of our cells have markers, and those markers are how cells communicate as well as be identified as belonging to you. It's your DNA that makes those antigens, and only you. So it's the immune system that is going to attack an antigen that doesn't belong in its blood. Type O is the universal donor, as I went over before. Type AB is the universal recipient. Now, the other topic that we went through pretty quickly, but there's not much you need to know. You need to know what it is, though. And in this case, the allele for the dominant trait is not fully expressed unless it has 2 copies of it. It has to have 2 copies or it does not fully express itself. So here is a picture of it. And we're talking about mating a homozygous red with a homozygous white. And you end up with the F1 heterozygous offspring. What has happened in this condition is you see pink. You need 2 red genes or alleles in order to fully express red. The lowercase r or recessive r is expressive of no color. That's why it looks white. And if the heterozygous plant has the lowercase r, which doesn't express anything, and the uppercase r is not completely dominant, you thus get a lowered red color. The bottom picture is showing you the cross of the F1 with itself and the F2 offspring. Remember, everything we do is parental, which is the purebreds, the F1 offspring of the purebreds, and then the F2 offspring of the F1 that were allowed to mate themselves. This picture is just another picture showing you the same thing. You got the P generation. You have the gametes that those two plants can make. Put together, they create pink because the dominant R needs 2 copies to fully express the red color. If there's only 1 copy, there's less red. Then when you do the F2 generation, just like every other one that we've done, they're heterozygous times heterozygous. You end up with 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So red flowers have 2 alleles for the red pigment. White flowers, they're considered mutant alleles. They don't make any pigment. Thus, when you put a dominant allele for red and a white or non-color or mutant allele, you end up with less red pigment. You need 2 alleles for red pigment.